Hello, my name is Ella Jenkins, and I have the CRAS protein with the G12C mutation. So we're just going to go ahead and get started. CRAS is short for Kristen Rat Sarcoma Viral Oncogene Homolog, which is a protein found inside of our cells in the cytoplasm in the inner side of the cell membrane, where it plays a crucial role in signaling transduction and helping cells grow and divide when they need to, but when it's mutated, obviously things can go very wrong. So this is my 3D model of my CRAS protein. It has a globular 3D shape with well-defined regions that bind to nucleotides like GTP and GDP, and they also interact with other signaling proteins. These are made up of alpha helices, which I have shown here with the amino acids, and then the flat beta sheets, The structure is important because it allows the CRAS protein to switch between being inactive and active, which is how they control cell growth. The primary structure is a linear sequence of amino acids that folds into a functional protein, and the secondary structure is, as mentioned, the alpha helices and the beta sheets, which provide structural stability. The tertiary structure, or the 3D structure, is the globular shape, so when they're all together they form more of like an actual sphere, almost kind of like a lumpy sphere, and it's formed by folding interactions like hydrogen bonds and hydrophobic interactions, creating the globular shape it does. As far as quaternary structures, the CRAS protein usually functions as a monomer, so therefore does not have any other subunits on it. But now we're going to talk about the actual G12C mutation, which I have shown here. Usually at position 12 on the protein, it is a glycine amino acid, but in the mutation it is replaced by a cysteine. Glycine is more of a small and flexible amino acid, which allows it to turn on and off normally, but then when it is mutated and switched to a cysteine, it locks the protein in the on state, which means that the cell just keeps uncontrollably dividing and growing, eventually leading to cancer. At the cellular level, like I said, this prevents normal regulation of cell division, so it's constantly growing and dividing, but at the organism level, it contributes to tumor development in lung, colon, and pancreatic cancers. Like I've said, the normal CRAS protein can turn on and off, which is a key function for healthy cells, so they can grow and divide normally and not become cancerous. But when the mutated gene is there, it is constantly signaling for cell growth, like all the time. So this is why CRAS mutations like the G12C one are found in many aggressive cancers. Now, while this model, I hope, helps visualize the protein, it does have a few limitations. So it is not to scale, obviously. So in real life, this is on a very, very, very small, or it is a much smaller scale in real life, is what I'm trying to say. Another thing about this is that CRAS, the normal protein, is constantly dynamic, so it's always changing shape, but with this, it's kind of hard to show. So just having a 3D model is more of a static model versus the dynamic thing that it usually is. And then lastly, this model also doesn't include the interactions with other molecules like GTP and GDP, which are essential for its function. So yeah, that is my CRAS protein model. Hope you enjoyed.